Thank you, everybody, for coming. Welcome to the Temporal Genomics seminar. My name is Brennan Reed. I'm a postdoc in Malin Pinsky's lab at Rutgers University in New Jersey, and I'm speaking to you from traditional territory of the Lenni Lenape people. This series is organized by the Research Coordination Network for Evolution and Changing Seas Temporal Genomics Working Group. If you want to learn more about our working group, we have a lot of information on our website. I will link that in the chat in a little bit. So this is a bi-weekly seminar aimed at showcasing research into temporal genomics. And by that we mean studies that use genetic samples collected over time, often historic and contemporary genetic samples, to examine questions that are relevant to the ecology and evolution of species over the past several hundred years, especially their responses to human-induced changes in the environment. And we're really interested in highlighting best practices and new methods for those of us who are interested in doing temporal genomics work. So yeah, we have a schedule up on our website, and this is the first of our 3 roundtable sessions with 4 early career researchers that we're really excited about. So they're going to be giving slightly shorter presentations on kind of a project that's part of their work. Note that we don't have enough time in the seminar series to highlight all of the amazing work that people are doing in temporal genomics. So we are planning a Twitter conference where even more people will have the ability to showcase their research and their ideas. Even if you don't have a fully formed research project yet, that's fine. We finally have some dates for that Twitter conference. It will be, we're planning on putting it on July 7th to July 9th, and we will have more information on that conference up on our website. We'll have a call for abstracts up soon. So if you'd like to participate, just watch that space. And just a few ground rules while our speakers are presenting. We ask you to please be respectful. Keep yourself muted, hold all questions until after the presentation is over. Once our speaker is done, we'll ask you to either use the raise hand button or indicate in the chat that you'd like to ask a question, and then either I or Joe or Katrina will be helping me moderate. So I think one of us will call on you if you have a question. And yeah, if we have anything kind of fishy happen in the Zoom, everything has been great so far for the first couple seminars, but we have an alternate webinar link. Something happens, then we'll do our best to email that link out to everyone and then resume in the webinar. So awesome, thank you guys. So all of that out of the way, I have the great pleasure of introducing the first of this week's speakers. So please welcome Dr. Joe McGirr. Joe received his BS in biology from the University of Colorado. For his PhD, he worked with Christopher Martin at UNC Chapel Hill, where he was researching speciation in pupfishes. And Joe is now a postdoc. Well, he's a postdoc in Andrew Whitehead's lab at UC Davis. He's been doing work there. I think he's got another exciting position that he's moving on to, but we're going to hear about some of his work in the Whitehead lab now. So let's give a warm welcome to Joe. Take it away, Joe.
Thanks, Brandon, and thanks for organizing. So I wanted to give a quick shout out to another working group that I'm a part of the Marinomics working group, and we're interested in best practices in non-model organism and genomics and evolutionary biology. We have a seminar series for panelists, getting a whole bunch of people together that are experts in different types of sequencing and asking them questions. And our first one is tomorrow. So I'll also post this link in the chat. Okay, so today I want to talk about using temporal genomic contrast to understand Pacific herring population recovery following the Exxon Valdez oil spill. So herring are a forage fish, and forage fish typically go through boom and bust population cycles, even when populations are unstressed. So these plots show changes in herring biomass for 2 populations off the coast of Alaska, here in Togiak Bay and Sitka Sound. And you can see that the population size decreased in the early 90s and then recovered shortly after for both of these populations. However, this wasn't the case for this population here in Prince William Sound in Alaska. Here, there was a population collapse in the early 90s, and the population size has remained really low for the past 3 decades. And there was a major event that likely contributed to this collapse. In the spring of 1989, the Exxon Valdez oil tanker spilled 11 million gallons of crude oil into Prince William Sound, and oil just covered the coastline, which was really bad news for herring eggs because spawning occurs on the shoreline. So it takes about 3 to 4 years for newly hatched herring to mature and join up with large schools in the open ocean. And we can see here that just a few years after the oil spill, this herring biomass dropped by 75%. So even though it's still hotly debated to this day, it seems likely that the oil spill contributed to the collapse. But what the real mystery here is why the population hasn't recovered after almost 3 decades. Every other fish population that initially declined has since recovered, so why not herring? Well, one thing we know is that they've been struggling with disease outbreaks. There are 2 main pathogens causing issues, VHSV and ick. These pathogens have plagued the population since just after the oil spill, but these pathogens aren't unique to Prince William Sound. Other nearby populations have likely been dealing with them for just as long. So given that the oil spill was unique to Prince William Sound, we think there's an interaction here between disease and oil exposure. So here's our hypotheses that might explain slow recovery in Prince William Sound. Given that half of egg biomass deposited in 1989 was within the trajectory of spilled oil and that this was likely a major selective event. And we also have some information from other systems where rapid adaptation to pollutants has favored these loss of function variants that impair the function of signaling pathways important for healthy immune systems. So in some killifish populations, Atlantic killifish, there are deletions in this aryl hydrocarbon receptor pathway in some of these genes. And these deletions allow populations living in really polluted areas to survive. So this aryl hydrocarbon receptor pathway is really important in being desensitized to survive in a polluted area. But this pathway is also important for healthy immune signaling. And then we also know that in 1989, the primary selection pressure was oil exposure, and then that shifted in later years to disease outbreaks. So our prediction here is that Prince William Sound might show a reduction in effective population size. And then we're thinking that genetic variation that enabled pollution resistance might also compromise immune function. And if this is the case, we should be able to see signatures of fluctuating selection pressures in Prince William Sound. So to get at this question, we have 1,100 Pacific herring, low coverage sequencing. And I just wanted to give a couple of very quick acknowledgements to members of the Whitehead lab that did all of the library prep, and all of the sequencing was done before I joined the lab. And then I've also been in a fun collaboration with Vince Buffalo, who's been looking at short-term estimates of effective population size. And I'll get to that in a second. So here's our really cool population sampling. We have this really nice data set. So for Prince William Sound, we have samples from 1991, 1996, 2006, and 2017. And these samples all came from adults. So the 1991 samples were not exposed to oil as embryos. So we can compare changes in allele frequencies pre and post oil spill. Another interesting comparison we can make is with this Togiak Bay population that didn't experience any oil exposure and is at a healthy population size. We have the same sampling scheme, so we can compare our post-collapse samples in Prince William Sound to this healthy reference population. And then we also have some more contemporary sampling across the Pacific coast down to California. So to test our first hypothesis and see whether effective population size was reduced in Prince William Sound, we measured the variance in temporal changes of allele frequency, which is proportional to effective population size. So this gives us a short-term estimate of effective population size. And using this approach, we see relatively stable population sizes in these healthy reference populations in blue and yellow. but an overall decline in population size in Prince William Sound between 1991 and 2017. Now the contemporary effective population size is similar in Prince William Sound to these other populations. So next I wanted to look for regions of the genome that showed shifts in allele frequencies consistent with this change in selection pressures. So in other words, I want to look for regions of the genome where allele frequency shifts change direction. And I'm going to do that by measuring this delta Z statistic across the genome in Windows. So for example, if there was a minor allele that increased in frequency between 1991 and 1996, and then decreased again between 96 and 2006, we would see a positive delta Z in this first comparison and a negative in this second comparison. So looking at that genome-wide, we can look at frequency shifts between 91 and 96 in Prince William Sound. And we see that there are these two really huge dips, one on chromosome 7 and one on chromosome 12. And if we look at the temporal shifts between 96 and 2006, that dip turns into a peak. So we can see that dramatic shift. So we can see significant shifts in the Togiak Bay population too, here in blue, but it's only in Prince William Sound in red that we see this reversal between these time points. So overall, this is consistent with the idea that some loci may have been selected for right after the oil spill and then selected against during later years when disease outbreaks were the more significant selection pressure. So this isn't the only region of the genome that shows this pattern, but it's very interesting because it looks like it's also an inversion. So when I measured FST between populations, I found these really sharp peaks of divergence despite really low levels of divergence genome-wide. And the inversions seem to be at a high frequency in the California population, the southern population, and then segregating at lower frequencies in these more northern populations. So my main takeaways here are, it looks like the Prince William Sound population shows a decline in effective population size between 91 and 2017. It seems like there's some evidence for temporal changes in allele frequency consistent with fluctuating selection pressures in Prince William Sound. And it's really exciting that inversions might play a role in slow population recovery. In the future for this project, we wanted to pair these analyses with measures of temporal covariance and allele frequency change to identify signatures of polygenic selection. So under drift, we expect random changes in allele frequencies across generations so that the covariance and allele frequency change between generations is around 0. But under directional selection, we expect a positive covariance. And with this approach, we could find stronger evidence for fluctuating selection and really nail down any potential genetic mechanism that might be contributing to slow population recovery in Prince William Sound. And if that sounds like fun to you or someone you know, then apply for my job. I recently accepted another job offer that I couldn't refuse, and it's really bittersweet because this is the coolest data set that I've ever worked with. But it's time to pass the torch on this project, so check out the ad for this job is on my advisor's website, Andrew Whitehead, and he has an absolutely fabulous group of people in his lab. And you'd also have the opportunity to collaborate with Vince Buffalo, who is pioneering new methods in temporal genomics. So with that, I would like to thank the Whitehead Lab. And I think actually we'll save questions until the very end. And we'll just have more of our roundtable discussion then. So I will set up for our next speaker. Okay, so up next, it is my pleasure to introduce Hernan Morales. He's a postdoc at University of Copenhagen. So take it away.
Thank you so much. Thanks for the invite, Joseph. It's a really nice opportunity to show the work we've been doing. Yeah, so okay, yeah. So as you said, I'm a postdoc in Copenhagen University, and this is really just kind of a couple of side projects that I've been working on aside to my main project. And one of my interests is like, what are the determinants of genomic diversity? And I have previously worked on more speciation genomics questions, so I spent some time thinking about this over longer evolutionary timescales. What is the relevance of certain genomic architectures to determine genomic diversity? and obviously the influence of demographic history. But I was really wondering what's the relevance of this over shorter timescales in the light of the ecological crisis that we are seeing. What is the effect of these recent but massive demographic events that some species have gone through? And if, as you just show, how the genomic architecture can matter a lot during these episodes. So for this, I'm running a project that is about measuring genomic diversity over time in endangered species. And really at the beginning was we had no pre-selected species to work with. So I reached out to a lot of very generous people that have been monitoring endangered birds for a long time, that have amazing samples archives. And we managed to set up the collaborations to work with 8 endangered species, 4 non-endangered as comparison. We are developing reference genomes for each of these species. And the idea is to do historical samples versus modern samples, and at least 20 resequenced genomes per time point per species. So this is quite nice because all these species have declined in different ways at different speeds. They have different historical demographies, so there will be different contingencies on these histories. Some of them have went almost extinct in the 80s, 70s. Some of them managed to recover through very successful management, and some continue to decline. So this is really just a comparative approach, trying to understand what is the genomics behind all these processes. And the approach was pretty much very simple: obtaining samples, empirical data, and then doing genomic simulations. But like about 1 week before we start getting samples, COVID happened. Everything closed around the world. So yeah, I shifted towards purely using genomic simulations for now. Well, now we're having samples and we're busy producing that data set. But today I'm gonna talk only about the simulation work. And I've been using SLIM to do all this work. And really the main features of SLIM is that you can work with entire chromosomes. It's individual based, so you can track every individual. It's forward in time, and you can simulate not only neutral variation, but also non-neutral variation. And there is very nice resources out there to work with SLIM. So the first example is, it's an ongoing work. So I've been using it to develop intuition in all these processes. And we really wanted to simulate, use the spatial features of SLIM to simulate the different types of bottlenecks that we can probably obtain in this data set that we're gonna collect. So we have one circular collapse, one peripheral collapse. I'm just speeding up because I have a lot to cover, and one that is random over space. So each of these dots are individuals, and each of these dots experience a spatial mate choice and a spatial competition around them. And they undergo these different types of bottlenecks. We also vary the severity that this happens, how fast it happens, and the dispersal distance that they can cover over space. So we have a lot of combinations of these parameters. I'm just gonna cherry pick a couple just to make a couple of points. So in this, we have 60% of habitat loss and the different types of collapses. You can already see that a random collapse is substantially worse. This is because individuals cannot find places to settle around them, and also the effects of inbreeding and drift in the sense of genetic diversity. Other thing that certain metrics we know are sensitive to population structure. For example, if you do nucleotide diversity across the entire population, you can get it really wrong. So this is part of developing the intuition. If you think you have substructuring, probably you want to look at your individuals first. And if you do nucleotide diversity over individuals, that will reflect a little bit better the true pattern behind it. And then just observing these things that have been pointed out by other people before, right? Like obviously when the collapse stops, that's where genetic diversity can continue to be lost for a long time. There is this genetic depth, essentially because inbreeding and drift keeps operating and actually become worse. or points that Debbie and that were made before that really think, where is your baseline, right? Where are you capturing your sampling when you're doing historical sample? Are you sampling when the population was already declining or at the beginning? And obviously the generations times, right? Like we see with the Galapagos tortoise. These slices are 20 generations. So yeah, probably if you have a very long-lived organism and long generation times, you're not going to capture a lot of these declines. And then we can also look to deleterious variation. This is really the power of SLIM, that we can model non-neutral variation. In this figure, we have heterozygosity on the left, and mutation load on the right for a low dispersal simulation. And you can really see how is this effect of when you have potential load containing an ancestral population where most of the load is contained in a heterozygous form and is not expressed. The bottleneck, what is really doing is exposing this potential load in a homozygous form due to inbreeding and converting it into realized load. That we can see here how load goes up over time. And when you have a substantial habitat loss, the effect is stronger and faster. Actually, here in the random scenario, the populations go extinct very quickly due to these inbreeding effects. But when we have higher dispersal, there is a rescue effect of across all scenarios. And this is because diversity is able to travel from place to place, heterozygosity is restored a little bit, and this potential load continues to be masked in heterozygous form and is not exposed into a realized load. So we can see very clearly that most scenarios don't express this realized load, except when you have substantial habitat loss. So we have been using simulations as well to test hypotheses. And there is, to look into these ideas that people have been putting forward lately, that is the importance of mutation load for conservation genomics. So if you imagine a space where you have low mutation load and high diversity here, where populations are going to be healthy, through genetic drift and loss of population size, they can move into a place where they will enter extinction vortex here with high, low, and low diversity. So we wanted to look at this with one of our test species, the pink pigeon, that have gone through massive habitat loss. They reached an historical low of 20 birds in the 90s, but through successful management, they were able to recover very successfully. Now there are about 400 birds in the wild. But through this bottleneck, they acquire a lot of nastiness, and they have pervasive inbreeding depression, segregating in the population. So we really wanted to use other capabilities of SLIM to do simulations that are very customized for the pink pigeon. So we change a lot of the life history parameters, and we simulate the precise demographic trajectory that the pink pigeon went through. So this is what has been recorded in the wild, and all these bars are captive population was established, and all these bars were translocations from this captive population into the wild population over time. So we simulated this through different scenarios of this genetic rescue, right? When there is no demographic growth and no genetic rescue, we actually observe over time the predicted trajectory towards the extinction vortex. Populations lose diversity and acquire mutation load, and they arrive to this part of the space. But if we do different treatments to this genetic rescue, we can have genetic demographic growth, but no genetic rescue that halts the loss of genetic diversity, but doesn't do much for the mutation load. And if we introduce new variation through genetic rescue, the process can actually be reverted. And if we select which individuals we translocate into the wild population by being those individuals that contribute as much diversity as possible and as little mutation load as possible, we can actually have gains compared to the historical population, reverting the process here to this zone of high diversity, low mutation load. And so the idea with this is like now we're busy generating empirical data of all these species and really use these simulations as a backbone to parameterize with empirical data the simulations, try to validate them with the same empirical data, and ask if we can replicate in silico what we can see in simulations, and ask if we can then do try to do some predictive work. So yeah, that was it. I wasn't keeping time, so hopefully I didn't went over time. Yeah, thank you.
Perfect, right on time, actually.
Perfect.
All right, very cool stuff. So now we'll set up for our next talk is Venkat Talla. He's a postdoc at Emory University.
Hi, thank you for organizing this. I'm gonna share the presentation. So I'm Venkat Talla. I'm a postdoc with YAB. First of all, can you all see my screen well and hear me well? Yeah. So I work on genomics of monarch butterfly population decline. On decline, I work on monarch butterflies, infectious diseases. I work on and the regular genomics of their gene flow and stuff. So monarch butterflies are this iconic American butterflies that migrate from United States and Canada to Mexico. But there are two presumed monarch populations in North America. One flies on the east of the Rockies and one on the west. So there are clear differences ecologically between these two populations. One flies to the west and one flies to the east, obviously. One overwinters in California and the other overwinters in Mexico. One takes a very long path to go from all the way to Mexico. The other goes much shorter, 700 kilometers path, and overwinters in California. One overwinters in the eucalyptus trees and the other overwinters in pine trees. On the west, they overwinter on the east, they overwinter in much higher altitudes in the big mountains. Most importantly, on the west, they are very low in numbers. They are only 1.4 million count. I think in 96, 97 was 0.4 million. While on the east, you have many more millions than the west. Approximately 400 million, the highest estimated count. When these were put on a flight mill in the lab, flight mill is just like a carbon rod on which monarchs are fly. This is a weightless rod, and we can record their flight information, the flight duration, flight length, and how long they can fly. So obviously, you can see one butterfly flying much slower, like a marathoner, but keeps flying longer, and the other flies like a sprinter, like it gives full bursts of power. So the eastern monarch, which fly longer, showed us they perform like marathoners. The west, they fly shorter but with a full power. So this is a clear difference in flight performance, and there are clear differences between the eastern and western monarchs. But the main question is it one population or are they two different populations? Our initial study using sequencing data provided by Marcus Compost from University of Chicago, we found we sequenced samples from the east and the west, and we found almost zero, like very low genetic differentiation, almost 0.001 average FST across the genome, and very low DXY as well. The divergence was also very low. And our demographic modeling using DADI also showed us that there's quite some gene flow happening between these two populations. But we also found two genes that we found that they have significantly different gene expressions. So these could be one natural population exchanging genes, but they have different gene expression in a few genes specifically to the ecology that they are set. So recently, monarch populations have been in use for their uh, enormously high population decline. So it's thought that 80% of Asian monarchs have been wiped out since '96, while Western monarchs declined more than 97%. Um, but these declines have been debated by some scientists because these declines were only found in the overwintering locations, but not in across the North America. So in the years where they found declining overwintering numbers, they found recovering counts from other parts of North America. When the North American Butterfly Association count butterflies up north in America or Canada, they don't see a drastic decline, and they see a very zigzag pattern than a consistent decline like shown in the Mexican population, the Mexican uh, overwintering site counts. So what are the cause? There's several causes that people think why monarchs are declining. One is extreme weather patterns. The other one is infectious diseases that they have. They have this parasite called OE, and they infect the monarch and they make the butterfly look very dark, and they don't hatch well. They have wing problems. Their fitness is very low, and the other reason is the main reason that a lot of scientists think is the introduction of GMOs and herbicide specific herbicide uses that kills the milkweed, and the monarchs won't have any milkweed to lay their eggs on. And the other, some other scientists believe that it's lack of floral food or habitat damage in Mexican overwintering site. So, are the observed declines in overwintering numbers resulting in a decline in genetic diversity? This is the first question that I wanted to ask. For this, we used museum samples like many recent studies as you guys were using. There are examples with bats, ibises, and recently honeybees that showed genetic declines with as a result of population declines that are happening in the wild. So for this, with the existing data set that we had, we added 1977 samples that were collected by the late Lincoln Brower that were stored in Florida Museum in Gainesville. These are approximately 140 to 150 generations apart, so that we can compare the contemporary genetic diversity with the historical genetic diversity and see if we find this population declines equals to genetic declines. So first, when we ran the population structure, the software initially told us that these are all one single population. There is no pattern. But we did the K-means clustering of 2 and 3. We found some individuals that are in historical samples that are close to each other than the contemporary samples. So, but the software in general thinks that these are one population. And then we went ahead and conducted genetic diversity calculations between within these individuals, and we found that the contemporary genetic diversity, nucleotide diversity, is quite somewhat a little bit higher than the historical genetic diversity, and the contemporary theta W, which is the Watterson's theta that accounts for the number of segregating sites, which gives you if a population is really increasing or declining. So if the theta W showed that the contemporary samples are much lower than the historical, we could presume that there are less individuals in general. But we found the individuals slightly going up. But and then the same result was found in Tajima's D, which is which is obtained when you subtract the theta W from nucleotide diversity. When you see nucleotide diversity and number of segregating sites increasing, and the Tajima's D more lower in contemporary samples than historical samples. That shows that the populations have been slightly doing better, or we did not see a pattern of a genetic decline in general. In the genetic differentiation as well, we did not see any extremely high genetic differentiation peaks between contemporary and historic samples. We found approximately the similar genetic differentiation between East and West and the historical samples versus contemporary samples. The site frequency spectrums also showed us the contemporary samples have much higher rare alleles in general in all site frequency classes or site classes across the genome, showing that there's some level of stability or an increase in the population. This is really surprising, as the natural populations have been reported to be plummeting. And you also have to keep in mind that the natural populations, the Mexican overwintering counts are not individual counts, but counts of area where the monarchs are overwintering. It's a count, hectare counts, but not individual butterfly counts, while the North American Butterfly Association counts are more of a butterfly count, people going out catching butterflies, counting. Declines usually, this is a very preliminary data that we got yesterday from a bioinformatician who is now working on simulating data to understand how much of a genetic decline do we expect in the Eastern and Western population. So in general, declines show faster in genetic data than increases. When population declines, a random number of individuals are removed from the population. So declines are showed faster, while increase needs drift and selection playing a role to increase the diversity. So our question
I
is,
just wanted to mention you're around 10 minutes now.
Oh, sorry. I'm done almost. Yeah. So why are we seeing these patterns? So enough monarchs might be migrating to keep the monarch population afloat. Conservation efforts might be helping because since the early 2000s, monarchs are heavily conserved. There's heavy conservation efforts going on for the monarchs. Declines might be more recent, and constant monitoring of genetic diversity is required for at-risk natural populations. Does the size of the winter colonies matter for genetic diversity? If yes, how much? In 2002, an estimated 80% of the winter colonies were killed, but the North American Butterfly Association count was very normal. So these are some important questions that we are asking, and we are sampling more to understand this better. With this, I want to thank my collaborators and supervisors and funding agencies, and thank you.
All right, thanks, Venkat. Definitely have some questions for you later, but we're going to move on to Sarah Gignoux-Wolfsohn, a postdoc fellow at the Smithsonian Environmental Research Center in Maryland. So take it away, Sarah.
Sorry, I know we practiced this, but I'm screwing it up. No. Oh God. Can you see that? Oh. Yeah, okay.
Yep.
That's good.
Great. That's not my first slide. Okay, thank you. Sorry about that. I think I'd have the hang of it after all of these many months of Zoom. Well, thank you so much for inviting me, and all of these talks have been super interesting and thought-provoking. I'm going to tell you a little bit of some of the work I did when I was a postdoc at Rutgers University, looking at bats infected with white nose syndrome. So you've probably heard of white nose syndrome. It's an infectious disease that affects multiple species of bats in North America. And since it was first introduced, it has caused massive population declines, especially in East Coast bat populations. This is a disease that's caused by a fungus, Pseudogymnoascus destructans. And what's super interesting about it is this fungus actually infects the bat, or is able to infect the bats while they're hibernating because their body temperature is lowered, right? We usually don't think of fungi as being a huge problem in terms of pathogens for mammals, but for hibernating mammals, they're actually able to infect them easier during hibernation. And the fungus causes lesions in their wings, and also, can infect their nose. But the way that mortality actually occurs is more indirect. This fungus causes the bats to wake up from hibernation earlier and more often than they normally would. And so when it was first seen, there were lots of sightings of bats flying around in the winter when they normally wouldn't, which then causes them to die of starvation or freezing. So it was first seen in 2007 in upstate New York, and has since spread throughout the Northeast. There have been sightings west of the Rockies. It's unclear if it is just less of a problem. It doesn't seem, the fungus itself has been detected west of the Rockies, but there don't seem to be symptomatic bats west of the Rockies, which is still a question. And it's not moving south because of that temperature limitation. So the good news, in spite of these massive population declines, is that bat populations do seem to be rebounding in some locations. So each of these is a different bat colony or hibernaculum, so usually a cave or a mine. And we see in red the colony size rapidly declines with the increase in P. destructans, so caused by white nose syndrome. But in the years since that rapid decline, for some colonies, there have been these increases in population size. This isn't uniform, though. In some hibernacula, we see that the populations are continuing to decline or have completely disappeared. So this project was started or was conceived of by my former PI, Brooke Maslow, who was thinking that potentially this could be an example of evolutionary rescue, right? So we had seen this population decline due to an environmental change, which in this case was the introduction of a novel pathogen. But now the increasing population size could be because these bats are evolving resistance and they will be able to repopulate. So in order to determine whether this could be an example of evolutionary rescue or an example of failed evolutionary rescue, aka extinction, she brought Malin Pinsky and me in to do the genetic work to see if there was actually selection happening in these bats that survived. So we collected before and after samples from what ended up being 4 different hibernacula. So in this case, the before samples were of non-survivors. They were bats that were actually collected and stuck in freezers after that initial mass mortality event. And then collected survivors about 10 years later. I'm going to be talking about these 2 populations, Williams and Hibernia, where we had both before and after samples. The 2 Vermont populations ended up being much further away than we could confidently say that they were before and after. So if we look at the population trajectories of Williams and Hibernia, this New York and New Jersey hibernacula, you can see that they both experienced pretty steep population declines. The Williams population started off as with a much higher population, but they both had experienced that population decline. Williams, we do see that the population is increasing. We see that kind of characteristic U-shaped curve. And in Hibernia, while the population remains low, it has achieved positive population growth. So these are two examples of colonies that could be experiencing evolutionary rescue. We did low coverage whole genome sequencing. It was about an average of 2x coverage across the genome, mapped it to the Myotis lucifagus. Oh, sorry, these are little brown bat samples. I didn't say that. So this is one species of bats, little brown bats. And there is a genome. It's very old and not great. There are people working on getting a better genome, but I mapped it to that genome and called SNPs and calculated allele frequencies using angst. So the first question is just how different are these populations across both time, pre and post, survivors and non-survivors, and also the different populations that we saw. So I calculated just genome-wide FST, and you can see that the FST values are very low. There's not a lot of structure between these populations, and there's not a lot of difference between the pre and post populations. And that was also reflected in this PCA. I then did some genome scans to look for selection. I ended up calculating allele frequency change and using that to look for selection. So I only considered loci or SNPs where there was a change in allele frequency greater than 0.5 in both populations. So really looking for parallel selection across the two populations. Obviously, there are other ways to look at this. I would love if people, wanted to follow up with this data and look for other signatures of selection. But this was kind of our first pass. And so to determine if these SNPs that had a high allele frequency change were significantly different or changing significantly and likely under selection, I did Wright-Fisher simulations and compared those simulated allele frequencies to our allele frequencies. And then to account for the uncertainty around the allele frequencies that angst calculates, I bootstrapped the allele frequencies. So by comparing the simulation to these bootstrapped allele frequencies, I was able to calculate p-values and determine which SNPs were significantly different and likely under selection. And I found that there were SNPs that experienced a significant change in allele frequency between the survivors and the non-survivors. And I found 63 SNPs that had this significant change in allele frequency, and so our candidate SNPs under selection. When I went into this project, I was assuming that we would see a hard selective sweep. So we'd see these big changes in allele frequency, and around that we'd see a big change in diversity or pi. In contrast, what I saw was a what we're calling a soft selective sweep from standard genetic variation, where there's no change in pi around our SNPs under selection. So I found that, I found these 63 SNPs, but there were no changes in pi or into GMAS D. So I have to suggesting that they are a soft selective sweep. But then also all of the alleles that were selected for were present in low frequencies in the non-survivor population, in the pre-white nose population. So we can conclude that this is a soft selective sweep from standing genetic variation. And these SNPs were located in a number of genes that were annotated. Going into this project again, I assumed that we would find selection in genes associated with immunity. because this is a disease. But as I mentioned, this fungus kills bats in a very specific and not necessarily classic pathogen way, right? So it kills them during hibernation. And contrary to my expectations, I only found one SNP in a gene that is associated with immunity. It's MASP1, which is part of the complement pathway, which is a part of the innate immune system. And interestingly, MASP1 has actually been found to be less abundant in brown bears while they're hibernating. So I think that it's less to do with the innate immune response and more to do with the depression of the innate immune response while these bats are hibernating, which then is allowing them to be infected. Two of the genes have been previously associated with white nose syndrome, PCDH17 and REPS2. They were found to be more expressed in bats that were infected, but only after arousal from torpor, which is interesting when you then combine it with us finding SNPs within these genes. And then finally, I found a number of SNPs and genes that are associated with metabolism. So when animals hibernate and when bats hibernate, they depress their metabolism, right? They're in this torpor state. And so I think, so it's interesting that we found these SNPs and these genes associated with metabolism, because again, it seems like what's more being selected for is that hibernation phenotype rather than classic immunity. Okay, so what does this all mean? I'm sure you all have seen these memes circulating on Twitter. This one is types of conservation genetic papers. And so, you know, my paper was basically, we found some small effect loci for local adaptation or GWAS. Let's base conservation policy on these. I'm obviously being somewhat facetious. I mean, this is, you know, a good first step. And there are people following up on my work and other work that's done similar studies in other populations of bats, trying to say, okay, so now can we take all of these SNPs that have been identified and look at their potential use with respect to policy, with respect to management, and also trying to tie hibernation phenotypes to genotypes. But one of the more interesting questions for me that came out of this work, and which I will pose to you all as part of our discussion, hopefully, is just thinking about how often adaptation to novel diseases is actually driven by stand-in genetic variation rather than novel mutations, especially with, you know, pathogens that are going to evolve much more rapidly than their hosts, that are being introduced and causing these mass mortalities. There isn't always time for what we think of as classic evolution. And so this is something that I've been kicking around in my head, and I'd love to hear your thoughts on. It's interesting with respect to Joe's presentation as well. Yeah, and with that, I'll acknowledge my co-authors and everyone who helped, and I think we're on to the discussion portion, so thanks.
Awesome, thanks, Sarah. Yeah, and I think I'll just jump right in with a question related to that last question while it's fresh in my mind. It is interesting that you're seeing signs of a soft sweep. So given that it's a novel disease, do you expect that maybe there were several haplotypes kind of floating around from past battles with maybe something similar that wasn't this exact pathogen, but maybe something closely related?
Possibly. I think what might be more likely is just that being able to hibernate well is something that animals are always going to be trying to, or doing a lot of, you know, surviving hibernation is important. And so I think there's probably a lot of selection happening and a lot of evolution of different hibernation phenotypes that then this fungus, because of the way it kills them, was able to select on. But also, yeah, I mean, it's totally possible. I don't know. I actually don't know how often bats are infected by other fungi.
Well, I definitely have more questions for folks, maybe while others gather their thoughts. I wanted to talk to Hernan and Venkat. We have until 9. How hard is that cutoff?
We can keep going. Certainly over that, if people want to talk, we've had discussions go, 10 minutes over before. So yeah, as long as people want to hang around and continue the discussion, yeah, feel free.
Cool, perfect. So one thing that we've been thinking about with this herring data set is maybe population recovery is limited by you had this big population decline, genetic drift is stronger, and then you increase your mutation load. And so just because there's a higher frequency of deleterious mutations floating around in the population, it's limiting population growth. Maybe Hernan, from your simulations, I don't really have a great intuition for how drastic that population size decrease would need to be. Like what that initial decrease would need to be for there to be an increase in mutation load. Is that something that you played around with your simulations?
Yes, I mean, I don't have a straight answer because obviously the results we have so far are very narrow into the parameter set that we have explored at this point. But I think it has to be quite drastic because it's easy for mutations to be just masked in heterozygosity. So you really need quite a bit of inbreeding for them to be exposed. And essentially, you can also have, if the decline is not as severe, you will have effective selection doing purging of these individuals, right? So you can actually have the ability to purge the deleterious mutations out if the effective population size is big enough and selection can operate. When you go really low, then you have a lot of genetic drift and selection is not effective anymore, and the population cannot escape. Not even purging can rescue the population. And that being said, in genomic architectures like the one that you observe, if you have a very long haplotype that is accumulating load, especially within an inversion, it will be more difficult to get rid of. Well, on one hand, more easy to purge it, but also more difficult to get rid of it once selection is not effective.
Mm.
So we do see, for example, we use realistic recombination maps underlying these mutations, these simulations, and we do see stronger effects in cold spots of recombination. So there is more diversity being lost in parts of the chromosome that don't recombine as much, which makes sense, right?
Yeah, that's really interesting. Does anyone else have any questions? Yeah, John?
Hey, yeah, great presentations, everyone. Joe, I had a question for you. I think you mentioned that you saw altered AHR activity in some of the herring populations, or potentially there was altered AHR activity, but are there any studies that have looked at like biomarkers of exposure and metabolism to PAHs and how that has potentially changed over time? And do you think DNA adducts caused by exposure to PAHs could increase the mutation load in some of these populations?
Yeah, so when I brought up the AHR signaling, yeah, I really condensed that summary there. I was trying to relate it to how we're thinking about it from another system in Atlantic killifish. There are these killifish populations that live in these really polluted waters. And when you compare polluted to non-polluted sites, the polluted sites, fish tend to have these really large deletions spanning genes in the AHR pathway. So one of the ways we were, and that desensitizes those populations to the PAHs and pollutants. So one of the ways we were thinking about in herring is maybe you had selection for maybe some deletions in these AHR genes, and that was great during the oil exposure. It allowed you to survive, but then later on, when you have these disease outbreaks, there's a cost to those same mutations. Now you can't mount immune response to these diseases. So it's still, all I've done is kind of look at AHR genes in windows, looking at those allele frequency shifts. And some of them do kind of show fluctuating selection if you really squint at it. But yeah, I think we'll have to use some other methods to really dig into that if it is a mechanism limiting recovery. I think Andrew had a question.
Yeah, hi everyone. Really great talks. It's wonderful to see such a great body of work from everyone. So congratulations, everyone. I had a question, that it was sort of brought up, and something that we think about a lot is the, standing variation, de novo mutation kind of contributions to these sort of rapid evolutionary changes. And I'm wondering if this is a place for simulation, because we just kind of assume that if you're a mutation that is saving a population from decline, it has to be standing variation, right? But maybe there's a place for simulation because, you think of a new mutation, it starts at very, very low frequency, and it have to be pretty extreme, strong selection for it to increase in frequency fast enough to rescue a population in decline. But how fast, how strong selection, maybe that's a place for simulation. Has anyone thought of sort of, maybe that's out there, I don't know. Has anyone done that or thought of doing that?
I haven't seen it, and I have thought of it, and I haven't done it. So if anyone wants to collaborate on that, I'm very interested in doing some simulations like that.
Yeah, I guess it really depends on the population size, as you said. Very, very low population sizes, genetic drift will take anything, any new mutations, probably. So I have the impression that does require a lot of a big selection pressure to catch. I haven't done anything related to that, but I have done a little bit of work with the fate of introgress adaptive alleles, and I have seen that it does require high, like very elevated selection coefficients for an introgress allele that essentially can, if you have a single introgression, it can be essentially something similar to a mutation, right? It does require a very high selection coefficient to catch.
I'll raise my hand and ask maybe a related question of something that I think about sometimes. In these evolutionary rescue scenarios where you do have this big demographic crash and then maybe recovery, you probably have, positive selection on these genes that will help you respond to whatever the stressor is. You also probably have, if the population is small enough, you're going to have those deleterious recessive mutations, potentially drifting to higher frequencies, potentially being exposed to selection. So you might have purging, and you might have an interaction. You could have linked, you could have those kind of alleles that are under kind of that standing variation that's under positive selection, but those could be linked to those deleterious recessives too. So I'd often wonder, and I don't know if you guys have thought about what the interaction in those evolutionary rescue scenarios is between purging deleterious recessive variation versus selection on the kind of adaptive variation.
Yeah, I mean, I think it's an interesting question. We weren't really able to look at it with our data, but it's something we're thinking about how to look at that.
Yeah, we've been trying to think about it through the lens of linked selection by using Vince Buffalo's method of just looking genome wide. If you're looking at recombination rates are going to vary. So if you look at temporal covariance and allele frequency change on the Y, and then your LD on the X, regions showing faster rates of LD should show higher temporal covariance. And I think there's a way that if you, because you're purging deleterious and selecting for advantageous, and that's going to have a lot of effects on your link selection pattern. So I think there's a way there to start to get at that. I had a quick question for Venkat. So you had really low divergence between your East and West populations. And then you had the one sample from 1977. Was that an East or a West population, or could you tell?
The 1977 population is an overweight population.
Sorry, can you say that one more time?
That is an Eastern population. It's from Mexico.
Okay. Yeah, so what population experienced the 80% decrease?
That's the Eastern population. That's 80% is the Eastern, 97, greater than 97% is the Western population.
Okay, so that's 80%
we
of
were
like,
unable to, oh, sorry. Go ahead.
sorry. There's 80% of like 400 something million, right?
400 million when people didn't actually count 400 million butterflies. So they assume for the amount of overwintering area, they assume there are 400 million. It's an assumption from the hectare count.
Okay. Yeah, so I'm thinking that's kind of similar with my system, where you can see this really huge decrease in population size without seeing much effect on your nucleotide diversity or some of those other summary statistics.
Yeah, it is the same pattern probably in like migratory species. Also, one best example is the locust. You see like this big outbreaks of locusts when the population increases drastically, but at some point they go down very low, like in relative to the huge population, they go maybe like 2% of when they were locusts, but you don't see their genetic diversity go up and down. So it's the same scenario, I think, when it comes to large migratory insect species. We actually wanted to get the same time point for the Western populations, but unlucky. We were very unlucky. Like we got Western populations from the 90s, but not before the 70s or even in the 70s. Yeah.
Yeah, Hernan, you got a question?
Yeah, it's just unrelated to that. I don't know if that's okay. It's for you, Joe, that you were saying that you are planning to do a polygenic scan in your data set. So I was wondering what's going to be your strategy regarding the inversion. If you're going to kind of like look at the collinear part of the genome, or you're going to also include the inversion, because I mean, it would be interesting to see what happens with the genome genetic content inside the inversion over time as well. But there is obviously link selection kicking in all over, right? You're muted.
Thanks, sorry. Do you see an acknowledgement slide or a signs of linked selection?
Linked selection.
Okay. So yeah, the way that we've been thinking about it so far is just looking genome wide, and then we're kind of assuming that those inversions are going to be lower on our LD here. But I think it would be really interesting to split out the inversions and run this analysis separate. Yeah, because really we're at a bird's eye view, just trying to say, can we see any signatures of linked selection if we just look across the whole genome? And it does seem like we see some signal in the Prince William Sound population after the spill, and we don't see that same signal in some of these reference populations. So yeah, I think we want to look now at, instead of Windows SNP level, and then it would be interesting to also just look at within the inversions.
Yeah. Yeah, I mean, you just expect a lot of correlation between loci inside inversion, right? So probably there's a little bit of bias. Or I was just wondering how much that affects the analysis, but yeah.
I see. Yeah, no, that is good to keep in mind.
I think Sarah's got another question. And Eric's got another question. So let's have two final questions, and then we can conclude. But yeah, Sarah, go ahead.
Yeah, I just was thinking, I don't know if this is a stupid question, but does anyone know, like, has anyone done a meta-analysis of how often there are population decline events where there's no loss of diversity? I feel like I see lots of examples of that, and our assumption has always been that there's going to be a loss of diversity. Has anyone looked at that across, or does anyone know of people who've looked at that across many systems?
I had the same question, but in terms of self-selected sweep versus hard sweeps, I'm just really curious to see which one is more prevalent. I was going to ask you, but we don't have the answer. Because it is similar work where I see a lot of evidence of small changes where there's small big changes, but a lot of small changes. So the examples that I see, they seem to be maybe more prevalence, bigger prevalence for self-selected, but similar question, but I haven't seen a review or something like that.
Right. Yeah, totally.
Same question.
I wish Debbie Lee were here, who did the first seminar, who was talking about, she did a big meta-analysis on historical studies where looking at whether there were changes in allelic diversity and heterozygosity over time. And I think the main point overall from that was, yeah, there's a detectable kind of trend in a lot of populations losing diversity. But I mean, there are also populations that have, retained diversity or that you would expect to. So I think it is definitely a function of, and her finding was, very isolated populations, island populations are the ones that you see those big expected declines in. So I think there's a big role of gene flow in this too, probably in butterflies and bats too. You'd expect, very mobile organisms. Even if you have these big bottlenecks, you might not lose that much diversity, but maybe in other kind of populations where you just have, no outside source of, no outside reservoir of genetic diversity, maybe that's where you see those declines.
Great.
I have a small thing to add there. So monarch populations, only the North American populations that are declining that people are saying are declining. We don't see a genetic diversity decline, but other monarch populations that are subsample of the North American populations, like the monarch populations established in Puerto Rico or Bermuda or Hawaii, all of them have lower heterozygosity than North American monarch populations, and those establishments were quite recent. So that's why we were expecting a genetic decline if there is a real population decline.
If I can add something, yeah, I was also wondering if the expectation is indeed to see a decline in a genome-wide diversity, because I mean, we know that very little migration can homogenize genomes very easily. But then probably the expectation will be something like what you observed, that there are like key parts of the genome that do have different dynamics. And that's why it's so important to really do genomics on this kind of stuff, right? Like that, I think that's my perception.
This is another spot where I think simulations can help you provide expectations for your data and for your species, because whether a reduction in nucleotide diversity is detectable or not is dependent on the nucleotide diversity you're starting with and the size of the bottleneck. And I think some like simulations can give you an intuition of like how much of a bottleneck would I need to detect a genome-wide signal given this starting level of nucleotide diversity, and it's surprisingly large. For killifish, I mean, they've got ridiculous levels of nucleotide diversity, but these populations that have evolved resistance in lethal environments, where almost certainly there was some initial, strong population decline, we hardly detect a reduced nucleotide diversity in those populations at all. I mean, it's detectable, but barely. So when you run those simulations, and Matt Osmond has done some of this. He was a postdoc at UC Davis for a while, now at University of Toronto. And, you need for anything with appreciable nucleotide diversity, you need a pretty wicked bottleneck to see it. So I agree, with some of these island populations, some of these really small populations to begin with, founder effects are real, but yeah, simulations, I think are recommended for lots of studies to be like, well, would I expect to see it anyways?
Okay, great. Thank you everyone for coming and asking questions, and thank you especially to our speakers. And I want to thank Joe especially because he's really been the kind of pilot for this session. I'm just the co-pilot. But yeah, thank you everybody, and we'll have May 20th as our next seminar with David Jacobs. Yeah, thank you everybody for coming. And please, if you're interested in the Twitter conference, there'll be info for that on our website soon. So great, have a great rest of your week, and hopefully see you in two Thursdays from now.
Hey, Brendan, I did have one more question. If there's one more minute, that question was for Hernan. So I really like your simulations with population. But correct me if I'm wrong, those were simulations for single populations. So I was wondering, so this is some stuff that sometimes I think about, because my previous system was fish populations along the coast. So gene flow was unidirectional. And so now we're starting to work in more systems that is more like a meta population where, you can have gene flow from many different populations constantly. So I was wondering if you had done any, maybe, I understand that it's already complex, and then there's going to be another layer of complexity, but I'm wondering if you've done sort of like simulating in a scenario, multiple populations with unidirectional gene flow or like a meta-population kind of a scenario, and how would that affect? Like I can see how it's going to affect, but I'm just wondering if you know, like one thing is thinking about it, another thing is actually running the models, right?
Yeah, absolutely. No, yeah, that's a very good point. No, we haven't done any of that because we were interested in this continuous space effect of genetic diversity. Probably there might be some insights into that with the random models, but definitely not unidirectional migration, but more like a meta-population outcome. But yeah, I mean, I do want to make the point that I do think simulations are very important, and this is just something that I diverted my project into for necessity. And Slim is super easy to use once you get the grip of it. So I do encourage everyone to use Slim, and there's a nice community, and Ben, the main developer, is amazing, and it's relatively easy to set up those meta-population simulations. So yeah, it will be cool to see it. Yeah.
Cool. Thanks, everyone.
Thank you, everyone. Bye.
Thanks all. See you later.